Hey guys, I'm Jillian Collins, or Jill is fine as well. I'm in the Chemical Engineering Department, and I'm a first-year PhD student here in Dr. Farnsworth Lab. Currently, our lab consists of myself, another first-year PhD student in the QBE department, which is Quantitative Biosciences and Engineering, as well as 2 undergrads are also in our lab. So we primarily focus on type 1 diabetes, and my research specifically is researching this protein called protein kinase C delta. And currently we are trying to study those mechanisms and how it could potentially be an important role regarding uncovering the mechanisms for type 1 diabetes, since the exact mechanisms are still unknown, which is why we're doing the research that we're doing. So today I'll be taking you guys on a quick little lab tour for Dr. Farnsworth's lab. We are still in the ramp-up phase since we are currently a new lab. However, we have most of it done, which is good. So to start off with, we do Western blotting in this lab, which is used to separate, identify, and quantify protein analysis. So the first step for this is you do what's called gel electrophoresis, which actually allows the separation of the proteins that we want to analyze. So we use these devices here where we basically inject them into these gels, and then we allow the current to run the gels, and that will allow the separation of the proteins. So what makes this unique about our lab is, as far as I know, we're the only lab that does Western blot analysis, so that is something that makes this unique. So this is what this whole setup is right here. Right here we have our -80, which is for stock storage as well as just freezing down cells for future use. As we continue over here, we just have stirs, weights, and we also have our fume hood, which is for synthesizing polymers. This is more of what our other PhD student, Meg, will be doing. She will be using this fume hood for her polymer synthesis for her project. So, to continue down the line, we have some centrifuges, and personally this is my favorite because it's a shaker, but it's called the belly dancer, so as you can see, it rotates around. So personally, that's one of my favorites because I'm just goofy like that. And then as we continue in this room, this is kind of our bio area. So for here we have our fridge where we keep all of our medias and other things. We also have a -20 in case the -80 is too cold for the things that we're storing. These are our biosafety cabinets where all of our cell culture work takes place. Alongside this, we also have our CO2 incubator, which is where we store the cells that we're trying to grow currently. And another thing that's neat about this lab is we have this nice microscope right here. And what's awesome about this microscope is that, so this cells right here in this culture flask, you can actually hook it up to the microscope and you can take images such as this one right here. So this is a current image that is taken from this flask, and as you can see, our cells are very confluent and growing nicely, which is really good. So that's one of the other pieces of equipment that I really like about this lab, because you can take these images for your analysis. We also have your typical water baths, and then we go this way here. As we go across the hallway, we have one more piece of equipment, which is the Western blot imager. Alright, so now we are in this room, which is where our Western blot imager is actually held. So once you do all the gel electrophoresis, you're going to take your samples, you're going to put them on a membrane, and then this machine here is going to read the data in terms of quantifying and analyzing your protein samples. So that's what this is for. And to conclude, the other thing that makes our lab very unique is, so although a couple of other labs in our department do work with animals, we are the only ones that have access or have the opportunity to actually physically do hands-on work with them. So you'll learn how to handle them, how to care for them, and do the proper procedures when you are dealing with the mice for your studies. This puts you at a huge advantage because when you go and apply for jobs, you can already say that you have that experience of not only working with animals, but also possibly designing your own animal studies, which will put you at a significant advantage towards other applicants for when you are applying for these jobs in the future, which is the whole point of getting your PhD in the first place. So with that, that is basically a brief tour of our lab, and unfortunately I wish that you guys could have come, but due to the circumstances, unfortunately you couldn't. So thank you for listening, and if you are going to be at the panel whenever we decide to have that this week, I will be there. So if you have any questions regarding campus life, student life, or our lab or other labs in general, I will be happy to answer your questions. Thank you.